My name is Rachel Hankin, and I am a second-year PhD student in Dr. Yao's lab at the Regenerative Bioscience Center. Today, I'm going to be talking to you about how I'm investigating the mechanisms of extracellular vesicle uptake into neural cells. Extracellular vesicles, or EVs, are lipid nanoparticles that are secreted by almost all types of mammalian cells, and they play a role in intercellular communication. Recently, EVs have gained attention in the scientific community because of their therapeutic and drug delivery properties, such as their ability to transport bioactive molecules and modulate the activity of recipient cells, as well as having low immunotoxicity and the ability to cross biobarriers, including the blood-brain barrier. Specifically, EVs derived from human neural stem cells have emerged as promising therapeutics for various neurological diseases. For example, in vitro and in vivo models, NSC-EVs protect against Parkinson's disease, as well as Alzheimer's disease, and improve recovery in stroke. When looking at NSC-EV biodistribution in mice, NSC-EVs are able to reach the brain, but most do accumulate in other organs such as the lungs, liver, and spleen. In order to improve NSC-EV biodistribution to the brain, my research focuses on understanding the specific mechanisms involved in EV uptake. There are three major steps of EV uptake. First, there is targeting, where the EV and the recipient cell recognize each other to initiate the uptake process. Second is entry, where the EV will enter the cell, usually via endocytosis. Third is delivery, where the EV will release its content into the cell. The step I am interested in is the targeting, and this is because EVs have been shown to have cell type specificity, particularly in the CNS. For example, Yao Lab has previously shown that NSC EVs will uptake more into mature neurons than human embryonic kidney cells. Also, EVs derived from oligodendrocytes are preferentially internalized by microglia compared to other cells in the CNS. However, the molecular basis for the selective targeting of EVs is poorly understood. My hypothesis is that receptor-ligand interactions between EVs and the recipient cells mediate the cell type specificity. Although various proteins, glycans, or lipids have been implicated in general EV uptake mechanisms, a specific molecule has not been identified. Therefore, in order to identify molecules involved in specific uptake mechanisms, we needed to develop a high-throughput platform to be able to screen for these molecules. In order to do this, we collected conditioned media from HEC293 T cells and subjected it to differential centrifugation to isolate the EVs. During this process, the EVs are also dyed with a molecule called CFSE, which gives them green fluorescence and allows us to visualize EV uptake into recipient cells. The isolated EVs are characterized using a flow cytometry-based method where we are able to measure their size and also quantify what percentage of particles are green. After seeding HEC293 T cells as the recipient cell in a 96-well plate, the CFSE dyed EVs are co-incubated with the cells for 4 to 6 hours. Then the fluorescence intensity of the recipient cells is measured to quantify EV uptake. The platform was validated using various methods such as a dose-response assay, using cold inhibition, and a time course study. As you can see, the fluorescence intensity significantly increases with increasing EV concentration. Also, when the EVs are co-incubated at 4 degrees instead of 37 degrees, the active EV uptake process is significantly inhibited. Also, over a 2-hour time period, there was no significant difference in fluorescence intensity across a 96-well plate. The platform was further validated by treating the cells and EVs with heparin. The drug heparin has been previously shown to prevent the EV's interaction with a molecule called heparin sulfate on the cell surface and therefore inhibits EV uptake. And our high-throughput platform was able to reflect this inhibition process. In conclusion, we have successfully established a high-throughput flow cytometry-based platform to sensitively and reliably measure EV uptake into recipient cells. Therefore, we can now use this platform to investigate the role that surface proteins and glycans play in the specific uptake of NSCEVs into neuronal cells. I plan to do this in collaboration with Dr. Weiss and the CCRC by using a CRISPR-Cas screening system to knock out molecules on the recipient cell and investigate their involvement in specific receptor-ligand interactions of NSCEVs with neuronal cells. If we are able to identify a specific molecule that mediates the uptake of NSCEVs into neuronal cells, then we can potentially engineer the EVs to increase their brain biodistribution and therefore increase their therapeutic efficacy. Also, this platform can be applied to many combinations of donor and recipient cells, such as cancer cells, to improve drug delivery or therapeutic potential. I would like to thank Dr. Yao and all of the members of the Yao Lab, as well as Dr. Weiss and Julie and Juan from the Flow Cytometry Core. Thank you for listening.